podcast, and today's interview is with Martha Carlin. Martha is a really dynamic thinker on a systems level and has created three different companies and organizations that are all focused on how the microbiome and the health of the microbiome interfaces with human health. The first is the Bio Collective, which all started out of her quest to help her husband with Parkinson's and genomic sequencing of the fecal matter of people with Parkinson's, and then later went on to develop a company called BiotiQuest that creates probiotics for this condition, but other conditions like metabolic health and sugar handling. And then another company that aims at soil remediation with bacteria, specifically to break down glyphosate. And so Martha's amassed a huge amount of information about the microbiome and how it interfaces with a lot of different aspects of human health. And in this interview, we talk about those things. We also talk about metabolic health in general. We talk about water and its flow in and out of cells. And we talk about the devastating effects of glyphosate in the soil. So thank you, Martha, for coming on our program to talk to us.
Thanks for having me, Jonathan. I'm looking forward to it.
Well, our mutual friend Morley introduced us, and you were just about to tell me how you got into the work that you're doing. You run the Bio Collective, which is a fecal biobank, and I'm assuming that your journey, there was something that catalyzed that journey into that work initially, because there's a bunch of offshoots of things that you've also accomplished in your years.
Yes. So actually, my background and training and coming out of school, my degree is in accounting. So people are like, how did you get to science? Well, when I came out of school, I actually went to work for Arthur Andersen as an auditor. So they teach you to question everything, examine everything for yourself. And they taught us a process for how to look at risk in a business called transaction flow review, where we would draw these massive flow charts of everything that was flowing through the business, looking for the breakpoints and interviewing people and asking questions about how those processes work. And in 2002, my 44-year-old husband was diagnosed with Parkinson's. And, you know, a diagnosis like that can be pretty devastating. I'm one of these people, like, I'm a problem solver, I'm a fixer, so I'm not going to take things lying down. And I looked at it and how they, you know, it's like, here's a pill, there's really nothing you can do, it's progressive. And I said, well, this is a systems problem. And so how can I look at the human system and see what's flowing through it and start to understand that? So I, of course, started with food, because food and water are the main things flowing through the system. And I started to teach myself what was going on with our food supply. There were some things. My husband had been, he was a marathon runner. So he ate a lot of like high carb, you know, the goos and things that you kind of boost that sugar metabolism. He had been drinking soy protein shakes every day for the 2 years prior to his diagnosis. So I started to look into the food supply and what we had available at that time, tossed everything out of the cabinet and tried to go organic. But in 2002, that was pretty difficult. And so I really started studying deeply what was happening to our food supply. And then along the way, just continued to build into more and more areas of the science and how that tied to Parkinson's disease. Then in 2014, I read a book called Missing Microbes by Dr. Martin Blaser. And he was talking about the rise of the use of antibiotics in human medicine, but also in agriculture, and how that correlated with the rise of chronic disease. And over the years of interviewing other people with Parkinson's, I also knew that many of them had had a childhood history of infection, strep infection in particular, and so repeated courses of antibiotics. And so that was kind of a light bulb moment for me. And I 6 months later, the first paper was published that showed that they could divide the two primary phenotypes of Parkinson's disease. One is tremor dominant, the other is posture and gait dominant, and those could be separated strictly by the gut bacteria. And I said, that's it. I quit my job and started funding microbiome research at the University of Chicago, doing time series whole genome sequencing of my husband's fecal samples and mine with a scientist by the name of Jack Gilbert, who was kind of one of the early pioneers in the craze of the microbiome. He had one of his children was autistic, and we were talking about the overlap in autism and Parkinson's. I was then introduced to Dr. Suzanne Vernon, who had studied chronic fatigue at the CDC for about 17 years. We saw some overlap there, and we said, well, what's holding back this understanding? Instead of calling things single diseases, perhaps these are metabolic issues within the microbiome, and we can accelerate the discovery of that by building a bank of fecal samples that we could do research on, but other researchers as well, and perhaps people would share data. Now, that part of the idea didn't work out so well. People don't like to share that much. But we founded the BioCollective and started collecting fecal samples and doing whole genome sequencing, looking for patterns and what might be going on metabolically across the population. And I was also looking to tie that back to the food supply and the genetic engineering of foods, because I had an intuitive sense that the microbiome would give us those signals to what was happening in the food. And so over the last 9 years, we built the microbiome bank, built some computational tools to see how microbes interact together, and then ultimately went on to build a bank of probiotic organisms for both human intervention and agricultural intervention to correct some of these key problems and bring those to the market. So,
wow, wow, that's fascinating. Well, I can see one of the reasons anyway that Morley lined us up. I did a series of interviews with people working in different types of regenerative agriculture, end of like Q3, Q4 of last year, and that really got me. That was a big paradigm shift for me in terms of not just human health, but sort of global, the health of the whole biosystem on Earth, and how crucial and how disrupted it has gotten, especially since industrial agriculture has gotten its foothold, for 120 some years, and how bad tilling is for the soil, because you're not providing the landscape for these organisms to exist. When you were doing your work, so let me just rephrase a little bit of what you're saying here so I understand it. You are looking at the metabolic health of the microbiome within a human.
yes, you're looking to see that it has the diversity of function to do all the things it's supposed to do: digest your food, deliver the minerals in active forms through enzymes, and provide short chain fatty acids, all of that. So as you start to lose function in the microbiome, then different aspects of disease will pop up.
And so is that a function of the loss of some organisms, a change in the composition of the microbiome with regard to genus, species, strain level events, because of all of these, like, all this messing around with the food supply? What This is a relatively new disease, Parkinson's, for humanity, relatively over our timeline of existence. So I'm assuming that the way that you saw it was there's all this messing around with the food supply that has caused a functional disruption in the microbiome.
So in Parkinson's, Parkinson's is complicated. There are a number of different factors, but what you can start to see in the microbiome is different elements of that. And in our microbiome research, we see signs of nitrosative stress, which ties back to certain microbes that are present and also not present, or functions that you've lost. That can also be tied back to chemicals in the food supply, and various assaults from that perspective as well.
Okay.
And actually that work from 2014, that first paper, there are literally thousands of papers now on Parkinson's and the microbiome. And, every quarter, a huge volume of new papers comes out showing this connection to inflammation. There's actually, there's an animal model of Parkinson's that's that LPS-induced animal model. And LPS is it's the cell wall of gram-negative bacteria. It's an inflammatory endotoxin. There's also an animal model in diabetes using LPS. And so what happens, you could get that, the leaky gut barrier and a higher level of endotoxin producing organisms. And then the LPS crosses that barrier and into the bloodstream. And depending on where it goes, it's causing inflammation and different disease pathologies, but Parkinson's being one of those.
What if you could sort of quantify the percentage of input to the emergence of Parkinson's, if you could quantify that as a percentage, what percentage would you assign to the microbiome disruption or health, and some of the other inputs as far as you see it?
Gosh, it's tough to put a hard number on it. And it's interesting because, you have also done a lot of podcasts about energy. And so there is an underlying fear-based fight or flight mechanism in Parkinson's, where there's a perpetual state of fight or flight. And that can cause brain chemistry that feeds microbiome chemistry, or you can have microbes that make the chemistry that then drives the brain chemistry. So it's this gut-brain interaction. And that's interesting. There was some research, I think it was around the 1950s, where they actually cut the vagus nerve for people who had some digestive issues, and people who had the vagotomy didn't get Parkinson's. So this communication between the gut and the brain was disrupted. So, I would say, it's a very large contributor, but it's still in the works of figuring out how all of the interaction of that ecosystem and the different assaults that happen over the course of life that sort of drive that. And one of the things I have done a prototype for, I'd really love to have, is we have a diagram that is sort of your life story on one page in a visual image that goes from your birth method to, childhood illnesses, different stressors throughout your life, what kind of diet you ate, did you drink a lot of alcohol, did you have job stressors, marriage, divorce, because all of these factors drive chemistry, but then that chemistry also drives what's going on in the microbiome, because that's where that interaction is occurring. And, one of the connections with Morley actually came from a Parkinson's person who said, you need to meet Morley Robbins because, he's got a piece of the puzzle that you need. And his deep understanding of minerals And iron in particular, because Parkinson's also has implications of iron overload in the brain and iron dysregulation. And so that just kind of launched this mutual love fest between Morley and me teaching him about the microbiome and him teaching me about minerals and showing that interface that goes on in the gut.
Yeah, that makes a lot of sense. Well, there's that I didn't know, but it's what's so interesting about iron is I feel like everyone that I am talking to recently has an understanding of iron toxicity in a vast number of pathologies, and it's like an old understanding that's like making a renaissance now in human consciousness, especially with healers. But there's also the copper, the copper component of that, right? I mean, because this it kind of stems out of the substantia nigra in the brain, so far as I understand, which is, the black substance that's the translation from Latin, which is melanin it's neuromelanin that is a copper dependent molecule. And so you need tyrosinase, which requires copper in order to generate that. Um, I wonder, have you spoken to, or I'm sure you're aware of his work, Dr. Arturo Herrera Solis? Are you familiar? You know
Yes,
him?
I have. I actually, it's been a few years since I've talked to him, but I've read his book on human photosynthesis, a number of his papers, and communicated back and forth with him a number of times. And actually, I found him because in our microbiome analysis, genes involved in photosynthesis were differentially expressed in the Parkinson's microbiome. And I don't remember exactly what that was today, but and I was asking Jack Gilbert about photosynthesis in the gut, and he's like, "Oh, there's no light in the gut." And I'm like, "Well, light can penetrate tissue." And so I started to dig, and I found Arturo Solis's work and started communicating with him and looking at that, and I mean there's so many connections to what's going on with melanin and the connections back to Morley and the food supply as well. Most people don't really have an idea of the different types of preservatives that are used in food, but tyrosinase inhibitors are used in food, and there's actually a new apple that they genetically engineered out the tyrosinase because that's what causes the apple to brown. And so, they want to be able to cut up apple slices and put it in a child's lunchbox and not have it brown. But enzymes are critical to the breakdown of foods and delivery of nutrients. So if you start to inhibit these critical enzymes, you're inhibiting a lot of other downstream important things.
So, between your discussions back and forth with Morley and minerals and the microbiome, and maybe on a physical level, this might be sort of the lowest common denominator, the ground state of function, but there is an even subtler energetic terrain below that is sort of adjusting for things. It sounds like what we're talking about is emotions, which is something that he talks quite a bit about, and I'm there with you. um, where I'd like to go is to get your take on how much of an impact and in what ways do you see the microbiome impacting mitochondrial respiration or cell respiration in the human host?
Well, the microbiome is what, I have this book called The Life Bridge, The Way to Longevity with Probiotic Nutrients. And it explains very well how microbes are the life bridge of taking inorganic matter and making it living matter. And so at that interface in the gut, the microbes are taking those minerals, making enzymes, making making the nutrients more bioavailable. And I use as an example in that book, it shows selenium. You know, if you just take the USP selenium, it's not very bioavailable, and you can take selenomethionine, I think, or
Right. Yeah.
that's a little bit more available. But if you take a fermented yeast, nutritional yeast product, the selenium in that is about 260 times more bioavailable for the body than those other two forms. And that's really the case with all of your minerals are going to be much more bioavailable to the body through that fermentation process. Most of your B vitamins are made in the gut by bacteria. A lot of people don't realize that. So if you don't have the microbes to make those B vitamins, you can't make energy. And copper, being one of those key minerals to energy, one of the things that Morley and I look at a lot is the organophosphates and glyphosate in our food supply. Glyphosate is a minerals chelator. So it is binding up minerals in the soil. It is binding up minerals in the plant. It is binding up minerals in our body. And the chelation constants are very strong for copper, which makes it not very bioavailable unless you can break down the glyphosate and unlock those minerals again for use. And so that, I think, is one of the key things in the microbiome, restoring the microbiome with bacteria that can break down the glyphosate without producing AMPA and enable access to those minerals and the production of all those vitamins and enzymes that are made in the microbiome. Then it can be delivered and the mitochondria can work again. And the same thing is going on in the soil. And, you know, we're looking at and making formulas that break down glyphosate in the soil to revitalize the soil microbiome for it to do its job and deliver minerals again as well.
So yeah, a couple questions from that. You mentioned that there's almost like a preference for copper, the binding that glyphosate performs. Are there minerals that it does not prefer to bind, therefore kind of increasing that amount in the food supply or increasing that amount in the gut?
There's a table of chelation constants, and actually I had the good fortune to spend last Thursday and Friday with Dr. Don Huber, who's one of the world experts on glyphosate. And he's supposed to be sending me a table. So I think it binds cobalt preferentially before copper. And Morley's really the expert on the orders of magnitude, but I do not think that it binds iron, or it does not bind iron at that same there is a preferential binding. And I think boron, manganese, and copper are some of the ones that it binds more strongly. And this is interesting too, because we actually did some research with Dawn looking at cabbage in a sauerkraut operation that was not able to maintain its structure. And they went through a lot of research to finally figure out that it was in an organic field, but they were using poultry manure, and the poultry manure had a high level of glyphosate, and that was binding up the copper, manganese, and boron. What little was there in the soil was getting bound up, and those are really critical for plant structure, and they're also critical in the human body for collagen structure. So if we think about all the people who are having joint problems and needing joint replacements, you know, it very likely goes back to the chelation of these key minerals that are needed for building collagen.
That makes a lot of sense. Yeah. So the product that you sell, that you created from the BioCollective as a result of banking all this and analyzing all it is Is that the product called Sugar Shift, or is this a different?
That is I made that product initially, like we weren't really thinking we'd be a probiotics company, but I went to a Parkinson's conference, and some researchers showed that the sugar alcohol mannitol could stop the aggregation of the alpha-synuclein proteins in an animal model and actually pull them out of the brain. And so I came back and bought this little mannitol chemistry book and started reading about this amazing molecule. It's a very powerful free radical scavenger. It's the most abundant sugar in nature. It manages osmotic stress. It is used for thermal energy storage. I mean, it has all these incredible properties, and Morley and I are looking at it because it's also used in scientific research as a media for mitochondria to keep them happy. So in that process, I found a handful. There's a small number of microbes that actually make mannitol from glucose and fructose. And so we came up with this 8-strain formula to do this conversion at a higher rate than any of the strains would do by themselves. We filed a patent for it, which we actually got awarded in December of this year. And at the time, my husband, John, was walking with a cane and really having trouble navigating crowds. And we were still measuring his microbiome. And he started taking it, and within 30 days, he was no longer walking with the cane. And we could see his microbiome profile was moving back closer and closer to the human, healthy human microbiome profile. And his UPDR score, which is how they measure your progression, improved. It dropped from a 35 down to a 20, and it stayed there for about 4 years until he had COVID. But in the process, then we started really looking at the mechanisms and sharing it with other people, and found that in that conversion of the sugars, it helps with blood glucose management, and went on to decide to bring that and some other products to market to address specific losses in the microbiome. And then we did a clinical trial in a diabetes cohort with the Sugar Shift in 2021.
Fascinating.
2022, I guess it was 2022.
So how does it interface with mitochondria, mannitol?
Well, I mean, when you're in a cell model, you need storage media, and that often is some type of a sugar. But mannitol crosses one of the membranes in the, I think it crosses the outer membrane, but not the inner membrane. So I think what we've looked at is it's shifting that membrane potential. And one of the things I've been talking to Morley about is it does complex with copper. So maybe it is delivering copper into the inner membrane of the mitochondria before it exits. Because most of the mannitol, if you were to eat mannitol, is going to be excreted. And it's mannitol's used. It's on the World Health Organization list of essential medicines. It's used to complex with a lot of different things. It's used for brain trauma. It crosses the blood-brain barrier and can help deliver things to the brain. So we're still kind of trying to figure out the details of how it might work with the mitochondria.
Yeah. So what are some of the other products you brought to market?
We have one called Ideal Immunity that has a specific strain that we found that is bacteria can make molecules called bacteriocins that are very specific. They will specifically target other pathogenic organisms, I'll say. So this one is 100% effective in our lab tests against Listeria. It's very effective against Salmonella, E. coli. So, we kind of say that's a nice one to have around for food poisoning, or if you're doing foreign travel, those types of things. And I have actually a physician in Hawaii who got something from one of his patients who came back from the Amazon jungle in the 1970s. And had chronic severe diarrhea until he found our product. He had tried everything, different, antibiotics, antivirals, different probiotics, nothing worked. And that was the first thing that worked for him. We have one called Heart Centered that helps with CoQ10 and nitric oxide production. We have one called Antibiotic Antidote that helps restore the internal ecosystem so that it can repopulate with a broader population of microbes. And we have one called Simple Slumber that helps with the production of bacterial melatonin and tryptophan for sleep.
Wow, that's really cool. So the immunity product, does this work in SIBO cases, or does it need to be in the large intestine in order for this to perform its action?
No, we actually think that the action in the small bowel is really critical. Particularly, I've done a lot of, I guess, collaboration with Dr. William Davis, who wrote the book Super Gut and is very focused on SIBO. He thinks about 50% of the US population has SIBO. So both the Ideal Immunity product and the Sugar Shift product have the microbes that can convert those sugars. And they do both make some bacteriocins. The Ideal Immunity has some very specific ones that Sugar Shift doesn't have. But I do have a lot of people in his inner circle who actually make yogurts from either Sugar Shift or Ideal Immunity in their kind of healing journey for SIBO. But people who have really severe SIBO and can't eat any FODMAPs or anything, they often have to do some initial work of cleanup before they can try a probiotic, because you've got inulin in there, which is a fermentable.
Got it.
So
Yeah, right. Okay, so it's not only these bacteriocins, there's other prebiotics in there that could potentially
right.
feed bacteria you don't want to be growing.
Right. So you've got to kind of balance that out. But I do have, customers who have reported that sort of cleanup process with those products.
That's fascinating. Wow. Okay. Um, in your work with Ancient Organics, which is the soil remediation, is it kind of working in a similar way?
Does work in a similar way. So, I was very blessed early on in the founding of the company to connect with Dr. Raul Cano. He was a professor of microbial ecology at Cal Poly for about 35 years and was a pioneer in using microbes to clean up oil spills. And in the process of working on our human probiotics, the conversation I had with Raul was, well, we can make all these products for people, but as long as we're still polluting the soil with organophosphates and hydrocarbons and all of this, we're fighting an uphill battle. We may never win that battle. So he had a collection of ancient organisms that he had isolated from a previous company. And he still had all of those organisms. And so we founded Ancient Organics to make these working systems for the soil to unlock those minerals, break down the glyphosate. But, for a farmer, they're less concerned about cleaning up their soil than they are yield. So, what we found in our studies is there's a dramatic increase in yield by breaking down, these toxic herbicides and pesticides in the soil, enabling the minerals that are there but trapped to get out and feed the plant. And so we've done tests in grapes. We actually in a vineyard in California where we showed reduction in glyphosate in the soil and in the grape. We just finished a cotton study where we showed a 25% increase in cotton yield in the fields that used our paleo power. We've looked at hemp and cannabis, vegetables, corn, soy, um,
Hmm.
kind of
Wow.
across the board. But it's also an uphill battle because you're fighting against the large agricultural interests who control the supply chain to the farmers. So, it's a little bit of a struggle, but I think people are starting to catch on. And Dr. Huber actually is a co-owner of the patent with us on that product. And now that he's seen our study results, is kind of over the moon. He's been waiting for this for 20 years or more.
Amazing. So is this only bacteria? Are there fungi also in this mix? Do you work with fungi?
Well, we're not specifically working with fungi, but actually, Raul, in his early part of his career, was a medical mycologist, so he knows a lot about fungi. Our mixture is bacteria; it's not fungi, but what it's doing is kind of in the same way ours in the human gut is. It's reshaping the terrain in a way with cross feeding that enables that better overall soil health and mycorrhizal development as well, because you're kind of opening back up the exchange of nutrients between bacteria and fungi.
Yeah, that makes sense. So, what got you interested in aquaporins?
Oh, um, well, so I had been looking at the Parkinson's microbiome in the context of something called molecular mimicry. And that is actually where bacteria produce little heat shock proteins that have a similar sequence to a human tissue. And so if your body is reacting to the bacteria and producing an antigen to that heat shock protein, if it's similar to a human tissue, it can cause autoimmune. And there were elevated antibodies to a number of these common, cardiolipin being one, which is the membrane of the mitochondria. But there were 3 different microbes that also produce cardiolipin. And so I was looking at that, and I came across a paper that was looking at aquaporins and autoimmune, and essentially showing so aquaporins they control the water channels between cells. They're highly conserved. Bacteria have aquaporins, plants have aquaporins, we have aquaporins. And I think we're now up to the discovery of 13 human aquaporins. It may be more than that now. But one in particular was this aquaporin 4 that controls the flow of water across the membranes in the brain. And in her paper, she was showing that I think it was spinach, tomato, corn, and I can't remember if it was wheat or soy, but had high sequence homology in their heat shock proteins to the human aquaporin 4. And so I started thinking about, okay, well, we know we shouldn't be eating corn and soy because of everything that's been done to that, but people think spinach is healthy. And so, we cut spinach out, and then I kind of ran off into a very deep dive looking at all these aquaporins and what is going on in the body with the delivery of fluid in and out of cells, because that's one of our, osmotic stress is a key factor, and it's a key factor in mitochondria. Mitochondrial swelling can cause damage to the mitochondria. So, it just was kind of another crazy rabbit hole area of interest to me, but had a tie back to the microbiome and what might be going on there and the signaling to the body.
Are these like enzymes embedded in the membranes?
They are embedded in the membranes, and there's a researcher in Australia that I have been talking to for the last couple of months, who is he's a physical chemist. He's writing about the glycocalyx in the endothelial surface layer, And one of the things have been talking to him about, because he has he has structural drawings of what that looks like, and he has these water channels in his structural images of how those membranes are built. And I think that those water channels that are in there are actually, these aquaporins, that dot just hasn't been connected to the physical chemistry images.
Have you spoken to Jerry Pollock about this issue? Are you familiar with him? I mean, it just
I'm familiar with his work. I haven't spoken to him, but I've read a lot of his stuff. There's a Korean scientist. His name's escaping me at the moment, because part of what I had also tying back to the elements. The Korean research. He has a book called the Water Puzzle, I think, and he talks about different elements being structure making or structure breaking, and that sort of piece of the puzzle to tying in the minerals and the conversations I have with Morley and what's going on with the water that makes water structure.
Yeah. Well, it seems like there is going to be a relationship with the quality of those membranes and how well that water is structured, and the electrostatic gradient that gets created as a result of which, light is a big part of that. And yeah, it got me thinking, because I think that is the same type of water that's created as the output of the electron transport chain. So this water flow and this is an area that's like, wow, it's like we're probably 10-20 years away from really understanding the gravity of how important this type of water is. Because right now we're just talking about ATP, and so it's like, okay, we'll talk a little bit about carbon dioxide. That's kind of important, but that you also have this water, this metabolic water, deuterium depleted water that's structured as an output of the mitochondria. And not only does it seem to provide a sort of vitality to the body, but also information to the body, which is maybe where that interface between emotions and chemistry occurs.
Very possibly. Yeah, a lot there. So we have also been looking at the microbes. So microbes need free water. So in that structure making and breaking, they need free water to do some of what they do in that interaction. But part of what we're looking at, and we've also been talking to Laszlo Boros, who does a lot of work with deuterium depleted water, is that the microbes are, potentially moving that extra molecule so that you are getting I think he calls it depleted water, but you're going from D2O to H2O, and all of that, maybe not all of that, but much of that may be actually going on in the microbiome. And, with the movement of those hydrogens. And back to glyphosate for a minute, it does disrupt that process in the microbiome.
Yeah, not good. Glyphosate. That's a big issue.
Yeah. And then sort of back to the dehydration concept. So when we started collecting fecal samples, one of course, Parkinson's was one of my main interests. So we collected more fecal samples from people with Parkinson's than other disease categories. But the people in our lab who processed the samples, so we would get a sample and process it into multiple little aliquots so that other scientists could access it. And they could identify a Parkinson's fecal sample without knowing anything about the person. They just from their experience processing fecal samples, it's like, oh, this is a Parkinson sample. We can't use our normal methods to aliquot it. They actually had to use a caulk gun because the stool was more like concrete, which again is a big indicator of a loss of water homeostasis and dehydration. And this is something I don't think, in neurology, they don't talk about that much. It's considered kind of a non-motor symptom, but this lack of understanding, there was nothing in the published literature about this either, which was kind of surprising to me, but that, chronic constipation can precede a diagnosis of Parkinson's by 10 to 15 years. So you've got this solid waste matter of bacteria that are fermenting and producing bacterial protein toxins that the immune system then has to deal with, and extracting whatever water they can. That's causing a pretty significant part of the issue, I believe, over time. And if we could start to understand that and address it way back here, maybe we can prevent people from ever getting Parkinson's disease.
How much of a role do you think calcium and potassium play in this water balance?
Oh, that's a huge, that plays a huge balance. And, one thing I can say, in the RCP with Morley's protocol, when my husband was having issues, if he would have issues this was at post-COVID, if he's having difficulty walking or, just kind of a little out of it, if he takes an adrenal cocktail, which is that, sodium, potassium, vitamin C mixture from the root cause protocol, he's like, or, he gets in the tub with Epsom salts where he's getting magnesium sulfate through the skin, huge difference. So, yeah, I think that's
Have you looked at parathyroid in the relationship of this balance?
I haven't looked at parathyroid per se. We actually did have a call with a clinical nutritionist this week who was looking at John's thyroid. And so in order for the thyroid hormones to work properly, you need tyrosine, phenylalanine, selenium, and there's one other element in there. And of course, glyphosate impacts the shikimate pathway in bacteria and plants, and that's where your aromatic amino acids are. So you're going to be disrupting that tyrosine phenylalanine that is needed. And it's interesting too, because in our, we would take questionnaires and history of people in our database, and many people, many with Parkinson's and many with some other disease down that had an earlier diagnosis of a thyroid problem. So to me, it's like maybe that's the canary in the coal mine of that first problem with the aromatic amino acids and the minerals chelation.
Yeah. Yeah. One of the things that I've been working on is understanding this dynamic of calcium potassium regulation, and part of it came out of my conversation with Robert Thompson. Do you know Dr.
I
Robert
don't know,
Thompson?
Robert.
Morley introduced us, but he is very focused on potassium repletion and a lot of this relationship between calcium and potassium. And for those that don't know, you want to have your calcium outside the cell and you want to have your potassium inside the cell. And if that's not happening for a variety of reasons, you're going to have some serious metabolic slowing and inefficiencies. And so that led me to parathyroid and understanding this relationship between parathyroid hormone, serum calcium, vitamin K2, A, D, magnesium. They're all interrelated in creating this balance to make sure the calcium's not inside the cell. And so I'm getting ready to do some personal testing on that to see where my levels are, just to kind of explore it more.
So another interesting connection in the food supply, most people probably have never heard of an ionophore. They are used in the agricultural industry in cattle to help them get fat faster. And, there's been sort of an uproar in the last number of years about the use of antibiotics in animal husbandry, and sort of lip service paid, I would say, to cutting back on that. But ionophores are somehow exempted from that. And so, I think that also might be implicated in the disruption of that calcium, potassium. And off the top of my head, I don't remember the exact mechanisms, but it is controlling the movement of ions.
Interesting. Wow. Okay. Wow. Um, Anything else you want to make sure we cover today?
Uh, I mean, my kids are always saying, oh gosh, mom, when then people talk to you and they're, oh, it's coming at us from here and it's coming at us from here and it's coming at, how do you wake up every day and be optimistic and hopeful? And, I am optimistic and hopeful because I know so much about the microbes that I truly believe if we start to foster a culture where we love our microbes and we start to understand how to build the right terrain, to have a healthy microbiome in our body, in our soil, in our whole world, that the microbes will clean up this gigantic mess we've made and build for us a beautiful world again that is healthy and vibrant. And so I am, eternally grateful to the microbes. I mean, my kids laugh at me too, because I talk to my little bottle of and I say, go help these people. And it's interesting on the frequency front. One of the species that's in there is a organism called Bacillus subtilis, and I have a paper about how that microbe sings. It puts out a frequency, and that frequency kind of knocks down some of the other pathogens. So I call it the singing microbe. So I think there's a lot that we don't see. We don't see the microbes, and then there's a whole energetic level on top of that. So let's keep a positive mindset and let's build a world where we love our microbes so they can love us back and help us be healthy.
Let the viewers know how they can find your products and in your websites and various forms of contact.
Sure. So you can find our probiotic products at biotiquest.com. That's BIOTIQUEST.com. I have a Parkinson's blog where I write about alternative therapies and Parkinson's in the microbiome, and that's called Martha's Quest. If you want to know about poop sample collection, we have the Bio Collective, and then our company, Ancient Organics Bio, is where we're doing the work with the soil microbiome and agriculture.
Awesome. Well, thank you. Thanks, Martha, so much, and good luck with all those things. And I'm looking forward to talking to you again if you're up for that around two.
I am up for it. Yeah, I really enjoyed it. It was a great conversation.
I'm hosting these retreats.